Hi, welcome to my presentation. I first want to thank the RTM-FM for giving us the occasion to present our work. My name is Volker Bäcker. I'm a bioimage analyst at Montpellier Ressources Imagerie. I'm going to talk about Napari-J, a plugin we developed at the facility. We use FIJI Imagerie for doing bioimage analysis, including for 3D and 4D data. We frequently use plugins like the 3D Imagerie suite, FIJI, LimeZack. Now, how can we visualize and interact with the 3D data and the 3D segmentation and measurement results? In Imagerie, there's a 3D viewer, but that doesn't meet our requirements. It's outdated and it's not supported anymore. There's also SciView, but we don't know how to use that, or maybe the project is not ready yet. And we found Napari, which is a Python software, and that looks and feels very good. It's easy to use. It handles 4D and multi-channel data. It has the potential to work with big data, and it's an active project. It also provides interesting plugins for doing segmentation, for applying deep learning algorithms, or for creating animations. Now it looks and feels good, but we can't open images with it. When we try to open UMI-TIFF images, the dimensions all come up in the wrong order. If we try to open ZZI images, that doesn't work. And if we try to use a bioformats plugin from Napari, that doesn't work either. So what if we could open images with FIJI using the well-proven Java bioformats and then just send them over to Napari by pressing a button? And if we can do that, why stop there? We also want to exchange annotations, measurements, and so on. Thus, creating combined interactive and automated workflows. In that way, we can combine the power of all the Imagerie microscopy plugins with the power of all these data analysis packages in Python. Okay, let's build a bridge between FIJI and Napari. It's only about 3,500 kilometers. How can we do this? We need to start the Java virtual machine from Python, and then we can use the package jpype, which allows to import and use Java packages and classes. Then we can convert the data on the fly. For example, for image data, we get the metadata from Imagerie, we get the linear pixel data from Imagerie. We will reshape the pixels into the right form according to the metadata, and we create a Napari layer from the image. Now, in principle, this is possible without copying the data, but we didn't really look into this yet. Okay, we also can talk in the other direction from FIJI to Napari, sending comments. And for this, we use JSON, which talks to a CPython, which has a Jupyter client. And this Jupyter client can easily talk with Napari's IPython kernel. Okay, that sounds complicated, but the complicated stuff is all hidden behind the Python proxy that we wrote. Okay, if you want to use Napari and our plugin, you need to have a FIJI installed. You need to have a Python, at least version 3.7, and it's also recommended to have Conda. To install Napari, if you don't already have this, you open a terminal. You need to have Python installed. You type pip install Napari all. That's all for the installation, and then you type Napari to run it. But it's recommended to do this in a separated environment so that you don't get conflicts of libraries between different software packages that you have installed. So you can create an environment with the conda create command. Then you activate this environment, and then you do the things I said before. Now when the graphical interface of Napari comes up, you go to plugins, install, uninstall plugins, and you to look for Napari-J in the list, click the install button, and that's it. Now the first time you have to do some configuration. You go to plugins, Napari-J connection after the installation, and you tell it where to find FIJI and where to find the Java virtual machine. Now you can check the automatically start button like this, or you don't have to manually start FIJI the next time you make a connection. There's one more thing that you have to do. You have to tell FIJI to use the J file chooser instead of the native file dialog, because when the native file dialog comes up, Napari and FIJI will both crash. But with the J file chooser, everything works fine. Okay, now we're done with the connection panel in Napari, and we can now, for example, open the image panel. And now we're ready to work with it. We can open images in FIJI and press the Get Image button to get them over to Napari. If these are labels, so labeled masks in Imagerie, then we can use the Get Labels button and we can send screenshots back and also create a new browser window, a new Napari window, and get other images into the new one. So this is how it looks like. We have an image in FIJI. We got it over with the Get Image button to Napari, and we sent a screenshot back to Imagerie. I'm now going to show you 3 little application examples in which we use this combination of FIJI and Napari. First is about 3D nuclei density analysis. The second one about 3D spot colocalization. And the third one is actually not about 3D embryo cell segmentation, but we have something similar, and we cannot show you this data yet. So to show you the technical aspects of this project, we use this embryo cell example. Okay, the first example comes from a context of cell adhesion and migration in embryonic development. We have a cell spheroid, and we have 2 other stainings. Here is red staining, or magenta in this image, and another one. And what we want to do is to find out if the nuclei are denser in the zones with the red staining than outside of these zones. The image analysis approach for this is classical. We use a log filter and a 3D minimum finder. It's a maximum finder, but we use it to find the 3D minima. To detect the nuclei, then we use a dbScan algorithm from the FIJI library, which we use via JSON scripting, and then we use another JSON script to find the nearest neighbors and measure their distances. And since we started this project before we discovered Napari, we add the results as additional channels to the hyperstack. So to visualize the nuclei we found and the nearest neighbors. Okay, but then without being able to see it in 3D, it's very hard to see if the results are actually correct. And also, of course, for communication purposes, we would like to show the things in an animation in 3D. And now, when we found Napari, then we could directly do this. We have this spheroid with a darkly stained nuclei and 2 other stainings, and We're interested especially in these zones with the red staining, which is shown here in magenta. So we can rotate and zoom in and out. So now we see all the detected nuclei. And as I said, these are not spots, it's just another channel, so another layer in Napari. Now we have only the nuclei in the zones with the high red staining. And here we have the clusters, the zones with the high density of nuclei, and the nearest neighbor connections inside of these clusters. Of course, our tool will report the distances in result tables, the distances within the clusters, and the distances outside of the clusters. Okay, that was the first example. Just let me show how easy you can create these animations. You just switch your scene to a view that you want to show, and then hit the capture button to add a keyframe. Then you modify the scene and add another keyframe, and then you just have to tell how many steps you want between keyframes, and you can save the animation as a video file or as an image series. Okay, the second example, we want to measure the distances between the red and green spots, but only in the area where there's not the nucleus and the cytoplasm, but outside of the nuclei area. So we first mask this nucleus area. Then we detect the spots, and we send the image and the spot coordinates over to Napari. There we can filter the spots by a quality threshold interactively with a slider on a histogram, and we calculate the nearest neighbor distances, this time between the two different spot populations. We also calculate Ripley's K function to have a point of comparison, what we would expect from random distributions, which distances. Yeah, and then we display the connections of points with a minimum distance that we can configure. So here is how it looks like. We have the image in Napari. Note that we have it as 3 layers, one for each channel. So we got over 2 spot populations from 2 result tables in Imagerie, and with each spot layer, there is a histogram that's associated to it, and with these sliders. So here we have the green spots, and here we move this slider up. So we will only take the spots which are above this given quality value. And now we can send only those spots back to a table in FIJI. And then we get the connections with the Get Lines button for all the pairs of spots between the two populations, for which the distance is closer or equal to a micron. So here we see a close-up of this. Oops, I think I went in the wrong direction. Yeah, we're done with this example. And the last little example is about using LimeSec for segmentation, for segmentating data that looks like this, where there are stained membranes. And it does a really good job with this. It's really amazing. We also use cell pose for this kind of data, which also works very, very well. And yeah, here LimeSec has its own 3D canvas. And it measures surfaces and areas, but we also want to measure other things like intensities within the whole cell or within the membrane, the cell morphology, and whatever our users come up with. And sometimes we also want to do some post-processing on these results in cases where the segmentation is not completely correct. Now, this is very puzzling, the situation, because we have very good segmentations, but we have the results in a form that we don't know how to use them. We found two solutions for this. The first one, we wrote a JSON script that uses LimeSec's API. With the API, we can access to the cell objects that LimeSec created, and we can create labeled masks from this. And then we can work on this labeled mask to measure and to post-process with MorphoLib-G. That works really great for this. And here you see how the labels look like in Napari. They don't look so nice, but this is because the resolution of this image is very low. Our actual data has a better resolution and looks accordingly better. Now, second way we can work with this. In LimeSec, we can export the cells as meshes. You can find this in this little LimeSec graphical interface. So this will create one folder for each cell, and within this folder we find a mesh file for each time point. And we can use PyMeshLab in Python to import this data. We wrote a little Python script, only 3 or 4 lines that does this and adds all the meshes, each one as a mesh layer in Napari. And once they are in Napari, we can do some nice things with it. We can group them, and then we can apply properties like the color, or if they are visible or not, to the whole group at once. Yeah, we can also, if we want, switch from labels to surfaces. That's not very difficult to do. Oh, sorry, I forgot to show you how it looks like. Here we can hide some of these outer cells, and then rotate the embryo a little bit so that we can see what's on the inside. And then we can show the whole group of cells again, for example. Okay, now we're done. Sorry. We come to the summary. So with our Napari-J plugin, you can currently get images from Imagerie to Napari with the channels becoming layers, and you automatically have the correct aspect ratio. Of course, only if you have the pixel sizes correctly set in Imagerie. You can directly get screenshots back to Imagerie. You can get labeled masks as label layers to Napari. You can get points from result tables. You can filter these points by the quality and send the filtered points back to Imagerie. And you can also get lines over to Napari. For this, you need to have a results table where the first 3 columns are the coordinates of the first point, and the second 3 columns are the coordinates of the second point. So the import of the meshes from LimeSec, we just have a little script for this, but we will soon add this to the graphical interface. And for the Python bridge, for the EJ Python bridge, which will allow to communicate both ways, we have a prototype that works very well, but we still have to do some work on it to get a proper release. Okay, let me thank the RTMFM again, and also all the people working on these platforms, Napari and FIJI, and creating all these great plugins and packages. And I also want to thank my small image analysis group, which consists of Leo Tellez, who worked with me on this project, and Jacine Ansil. Thank you. We're ready for your questions.